Okay, yeah, I'll give a short introduction to myself and what we've been doing. So my name's Ken Young, and I'm based at the Royal Surrey County Hospital, and I'm essentially an imaging scientist, or a medical physicist to be exact. And I've been running a centre in Guildford for about 25 years, advising the breast screening programme on the technology for breast cancer imaging. And perhaps more relevantly, for the last 6 years, I've had a Cancer Research UK grant called Optimam. And in that, we're trying to answer some of the problems that Dave suggested in the mammography area, in that we've been building a very large curated mammography database, which is anonymized, designed for research, and that we're sharing with other researchers. And at the moment, we have about 67,000 images and 3,000 annotated cancer cases. But in principle, our initial idea was to create a national database, a bit like David was suggesting, where all the screening centres would contribute their images to a research database. But at the moment, we get all of the cancer images from 3 centres in the south of England. And we are just beginning to do some of the research, such as applying deep learning to the images and more application of more traditional classifiers to apply machine learning to identifying cancers in the image. Okay, that's probably enough for me.
Thank you.
So I'm obviously being Patrick for this period of time. I'm Sofia Olhede, so I'm a professor of statistics in the Department of Statistical Science, also with an honorary affiliation in the Department of Computer Science and one of the directors of UCL's Big Data Institute. I think what makes imaging so exciting to a statistician to look at is all of the possible forms of variability we can see in data and all of the dangers that can happen when we start to sample data across populations and across different imaging modalities. So there's a rich sort of degree of structure that we can get out of the data, and the question is, what information can we actually find in this sets of data? So I've worked on many different modalities, going old-fashioned EEG all the way down to more exciting sort of multimodal imaging projects that are more recent standing for me. And I think what's really exciting here is just the potential of what we can discover.
Okay, my name is Stuart Gibson. I'm an academic at in the School of Physical Sciences at the University of Kent. My background is physics, but like many physicists, I'm a versatile animal and have published work in all sorts of different areas, including chemistry, forensic science, actually quite a few psychology publications. There is, however, a common theme to my work. That is, well, if I had to pigeonhole myself, I'd say that I was probably an image processing, computer vision, stroke machine learning kind of a person. So as well as my academic role, I'm also a director of a university spin-out company. We sell facial identification software, or develop and sell facial identification software to, in fact, the majority of UK police and also to many law enforcement agencies around the world. Our system, not surprisingly, is also based on PCA. We've heard a lot about PCA already today. And we also use something called an interactive evolutionary algorithm to develop a facial likeness to a perpetrator in a crime. Patrick mentioned compressive sensing. This is something that we've also looked at, but only in brief. And in fact, we have a little schools project to develop a single pixel compressive imaging system. And we developed this system for the pricely sum of £50. So it's nice to know that you can do cutting-edge science on a very limited budget. Like Patrick, I think compressive sensing is likely to make an impact in bioinformatics. Yeah. Okay, thank you.
Great, thank you. So I'll open up to the floor. Keith?
So I must say I greatly enjoyed the talks. Could I ask all of you, I'm a doctor, been a doctor for a very long time, and I observed the development of medical imaging from looking at sort of hazy tomographs through to the extraordinary precision of modern CT. But you have the advantage that you are dealing with essentially digital information. And the great challenge, I think anyway, in clinical medicine, is taking the techniques that you've used so successfully and applying those to the, as it were, the clinical narrative, which is not digital information, is much more complicated, and where, on the one hand, you're in danger of articulating the obvious, which is what goes on in clinical trials, as you've described. You set up a study in which you identify some things in advance, which are absolutely straightforward, and you see how they relate to the treatment, to the investigations, or fast forward to the genomics, proteomics, metabolomics, etc. Whereas what is much more interesting is the sort of absorption from this inchoate information, stuff that you don't know, in order to relate it to things you don't know. And I just wondered if you've got any ideas how that might be done.
Who wants to tackle that one?
Well, it's a very interesting question. So in a sense, the problem with images is that they don't map straight onto a diagnosis, and it gets filtered through our experience as well at looking at the images and deciding what do the different shapes we see in the images actually mean. And if we take different types of information, like where people live or what their previous history was, you can't in a simple mathematical way just squeeze everything together to a summary measure. So yes, it's a difficult problem, and I think it has many components ranging from trying to understand how people's experience in seeing images after having done clinical diagnosis for many years interlink with how we're used to using technologies, right? Because our technologies output something for them to look at and get used to looking at, and then trying to combine it with different forms of data. So it's very much a big data problem because we have many different forms of information and it's increasing. But at the same time, it comes back to fundamental problems, which is how do you produce a model that can predict something, and what should you be calculating to answer the inference question you actually want to tackle? So it's an old question in a new guise, and there's no trivial answer.
Can I just say something? So just in our research, actually, I'm based in a hospital and work very closely with clinicians, and we have 3 eminent radiologists on our steering group. And in fact, as you say, what actually matters in breast screening, which is my area, is the decision that a radiologist makes when he looks at an image. That's the ultimate compression. He looks at an image and he says, I'll recall the patient, I won't recall the patient. So our approach to linking to that is that what we generally finally do after lots of research is present images to radiologists in a controlled experiment which mimics as closely as possible their normal clinical situation. And we ask them to make their normal clinical decision based on images that we've manipulated or distorted in some way. And our ultimate measure really is what's the quality of the decision making by the radiologist. And that's your ultimate test, because for quite a long time to come, we won't be allowing machines to make decisions on their own. The very best they will do is provide some additional information for a radiologist. And I think at least that's our approach to dealing with this issue.
So first, I think that was a very good question, because the different types of information and how they're used is key to the to looking after the patient, be it diagnosis, screening, or whatever, or deciding what the therapy should be. I like to think that the work I described from Danny Alexander's group on the Alzheimer's is at least a small step towards trying to sort that out, because that is taking data of very different types, information of very different types. So although the cognitive tests are turned into a numeric, the cognitive tests start with a sort of question, a Q&A process with the patient. Now you can argue about whether the turning that into just a number or a set of numbers that goes into a program that we might write, whether that's valid, and I think probably more work needs to be done on that. But it does at least show one way in which one can get the very different types of data together, in that case, to try and understand more about the disease progression. So it didn't have a therapeutic aim. It was trying to understand the disease progression in order to be able to then make some decisions, because you appear to have 2 or 3 different groups.
But if I may respond, those tests were, again, tests that were, although subtle, they were still obvious. And the question that I think I'm addressing is, how do you identify things that you don't know about rather than those that you do know about? You alluded to this in your closing remarks about data-driven stuff versus hypothesis-driven stuff.
Right, okay, so learning from the information's out there.
And so a rather naive hope that people like me who know nothing about this is that machine learning will reveal things which unmachine-like people like me won't actually.
Well,
We're all machines.
Maybe I can give an example. One of our approaches to looking at that is to look for interval cancers. So, or in fact, when a radiologist identifies a cancer in one of our digital images, we can look back to the previous screening mammogram, look at the same location and say, was there something in that mammogram that the radiologist could have seen? And what we actually find in the breast screening program is that in about 25% of cases, there is something that a radiologist can say, yes, in retrospect, either I missed that or I misinterpreted it. And so it's a task for machine learning to see whether you can identify could a radiologist plus a machine have done a better job when he saw that original feature that he didn't recall as a cancer. So that's one way of finding something that we're currently missing.
well, what's very much hidden there, right, is your own utility and depending on what different decisions you make, right? So the danger of trying to discover hypotheses from data is how do you make the test to be the right degree of sensitivity? Because if you do enough searching, you're going to find a lot of things that really mean nothing, right? So it then comes back, is it better to discover some things that are actually not present so you can do more tests? Or will those tests themselves be of damage to the patient and something you don't want to do? So as a card-carrying statistician, I always prescribe some degree of caution when trying to discover hypotheses from data.
Well, remember what Charles Darwin did on my closing remark.
Yeah, Dane.
So, I have two questions. They're actually related to the first set of questions. Again, interpretability. So in the talks, basically you have images, you extract some conceptual features from the images, 440 features, texture and whatnot. Has there been any effort to say, okay, this is a predictive or important feature, this particular weird texture-like feature, what does it mean at the cellular, molecular, morphological level? Can we understand why this feature is so predictive? Is there any work on that? And the second question is really, I mean, again, in my own work in single cell, I've started looking at sort of molecular single cell imaging, and have you people also begun looking down to cellular and molecular resolution in your work?
Okay, so you say about our people. Of course, we're a large community and only 4 of us here. So a lot of different people are doing lots of different things. So this is my take on the texture stuff. I find it somewhat frustrating to just come up with a whole load of different texture parameters and then throw them into the sort of algorithm that I described just now. I think it's much more interesting to understand what's happening with the biology. So, for example, some work that we're doing is to try and understand the MR diffusion signal. I didn't have time to talk about it because that is related to the cellular structure. The MR diffusion proton you can think of as being a little, almost like a measuring tool at the micron scale, although your readout is at the millimeter scale. And so there's some work that, in fact, we're doing with Danny Alexander in the back there to try and understand tissue microstructure and to relate it back to something about the biology of the tumour as it's growing. So yes, there is work going on. I wish there was more, but that's one example of something that we're trying to address. So I think there's widespread realization on both sides of the fence. Actually, I don't think there is a fence, but you know what I mean. The sort of cellular biologists through to the medical people. And there is, at least on the technical side, work underway to bring these two communities together better. I think they've evolved somewhat differently in the last last 20 years. And I think for at least the last 5, there's a real push to get people to use the same tools, to share tools. The open source paradigm, I think, has helped that a lot, so that it's easier for us to share tools. So I'll do a plug for somebody who I don't think's in the audience, Jason Swedlow up in Dundee, who I think has done a fantastic job at trying to bring the, he's coming from the life science community, to bring the communities together in the UK around the Bioimaging UK initiative, and there's the Euro Bioimaging initiative, which I know Heidelberg are heavily involved with. So these are attempts to try and bring the community together. I think CRUK has an important role here as well, and I think their recent funding announcements are going to help to make that happen.
Just wanted to ask a little bit about the role of the human observer in some of these things. And it's because it's a two-way process. In many ways, the best search algorithm we've got at present for most imaging is still the human eye and brain. That could be, for instance, an radiologist who's very good at spotting cancers. How do we feed that back into a sort of a learning about what they are spotting? Is there any work underway in that way? For instance, in cardiovascular disease, there are known cardiovascular experts with very good ears for murmurs, and they've been used as a probe to discover what they're listening for. So that one way. And the other direction is, what can we do with this information to drive better med-ed? Because clearly there's a variation of practice out there about image interpretation. And how can we use the sort of stuff that you're generating, the data sets you're generating, to drive from the algorithms back to better practice?
Can you've been very quiet.
Yeah.
That's about the human
I think
eye.
for good reason.
Yes,
I don't actually work in
yeah,
medical imaging.
The human eye stuff. Yeah.
So that's an interesting question, which basically means I don't know the answer. Yeah, I mean, I guess machine learning can inform human judgments of but both image quality and image diagnosis. So there are supervised learning methods in machine learning, and there are unsupervised learning methods. And it's possible that unsupervised methods might locate something in the images which was not expected, and then an expert could view the image and learn from that. I don't know if anyone else would like to add anything to that. But I guess that would be one way in which machine learning could inform the human expert, so it could be a two-way thing.
Maybe I can say, as I mentioned before, we use radiologists. We work very closely with radiologists. So one of the things we do is we do a controlled experiment. Typically, we might have 6 or 7 radiologists will review, and they don't all agree with each other. There's huge variation between observers, which is interesting in itself, but they actually perform very well. They are the best observers. And we took those images and ran them through a commercial computer-aided detection system widely sold and used in the United States, or not in the UK. And actually, they correlated that very, very well. And the thing that we were interested in was how changing the quality of the images affected the radiologists, because as a medical imaging scientist, I'm very concerned with quality. And we found that as we lowered the quality of the images, the radiologist performance dropped quite dramatically. The extra noise in the image caused them to miss small features which affected their performance. And exactly the same thing happened with the computer-aided diagnosis system. Although the manufacturers told us we couldn't assume that wouldn't happen, but actually it did.
So, but presumably those systems attempted to denoise the images prior to segmentation.
I'm not sure they do, actually.
Yeah. Okay.
One of the things is a major stumbling block that we have for this kind of research is, at the moment, the NHS in their image storage system tends to throw away the raw data. And any image analysis computer learning algorithm requires the raw data. In order for us to do our research, we've had to change the practice and stop them throwing away the raw data, because you can't. And in actual fact, I think Cancer Research UK could fulfill a very useful lobbying exercise to actually encourage an attitude of not throwing away useful data and also storing data in a sensible, organised way, which doesn't always happen.
I think here...
Patrick is itching to comment.
I just wanted to chime in on the initial question from the machine learning perspective. So one of the sort of fathers of modern machine learning theory or statistical learning theory is a fellow named Vladimir Vapnik. And I know from talking to the deep learning people, I talked a little bit about deep learning, that Vapnik at the moment is working on a paradigm where a machine learns from a teacher, but then you can ask yourself if it's mathematically possible for the machine learner to eventually exceed the performance of the teacher. So that could be something that's very interesting here. It's a little bit hard to visualize in this context, but if you think about something like a tennis coach, right? I mean, a tennis prodigy will work with a tennis coach and then rapidly, exceed you'll end up beating your coach at tennis or chess or whatever. So that's something that could be on the horizon, sort of further downstream, maybe in 10 or 15 years. But I think there's something interesting there to think about.
But it also brings back the paradigm if you're building the model to perform well for prediction, or if you're building a model to understand the system you're observing. So if you're building a model to understand the system, and if you're doing things for vision, you might be using features that are interpretable as being rough or smooth or associated with certain frequencies, then you can explain it back to a human being and tell them what to look for. But if you're using something like deep learning, which currently cannot be interpreted in such a way, then it's a lot harder to replicate that. So sometimes you just want to do engineering tasks, you just want the system that performs well, and sometimes you want to understand the underlying principles of the system you're observing. So I think there's room for both approaches, but not all are equally transparent in how they work.
I think that's an excellent way to end this panel. And let's please thank the panel members and to this afternoon's speakers.